It will be helpful to review the heart anatomy before we begin with the cardiac cycle. Recall that there are two receiving chambers. These are your two upper chambers, the right atrium and the left atrium. They're receiving blood from veins. The right atrium is receiving blood coming back from the tissues by way of the systemic circuit, and the left atrium is receiving blood returning from the lungs by way of the pulmonary circuit. The two lower chambers are the ventricles. They are the ejecting chambers. What this means is that they pump the blood out. The blood moves into arteries. The right ventricle pumps the blood into the pulmonary circuit, taking the blood to the lungs for gas exchange. The left ventricle pumps the blood into the systemic circuit, taking the blood to the tissues. There are four valves, two valves that are between the atria and the ventricles, and these are called atrial ventricular valves, or AV valves. The right AV valve is between the right atrium and the right ventricle. Another name for it is the tricuspid valve. The left AV valve is also known as the bicuspid valve, and it is between the left atrium and the left ventricle. The second type of valve is called a semilunar valve, and there are two of those. The pulmonary semilunar valve will be between the right ventricle and the pulmonary trunk artery, which is part of the pulmonary circuit. And the aortic semilunar valve will be between the left ventricle and the aorta, which is the main artery in your systemic circuit. Let's look at blood flow through the heart. Blood flow is unidirectional, meaning it's going to go in one direction, and the valves are actually going to function in that aspect. So they're going to make sure that the blood only goes in one direction and you don't have any backflow. The heart is going to function as two separate pumps. So we have a right pump and a left pump. The colors here are going to indicate the level of oxygen in the blood. Blue is going to represent low levels of oxygen, and red, high levels of oxygen. Let's start with the right side. The right side of the heart is receiving blood returning back from the tissues. That blood is going to be low in oxygen because the tissue cells have been using that oxygen to carry out aerobic cellular respiration. And we're going to have higher levels of carbon dioxide, which is a byproduct of aerobic cellular respiration in the blood returning back to the right side of the heart, compared to the blood that is in the left side of the heart. Let's see what happens to the flow. So blood returning from the systemic circuit is going to empty into the right atrium. There are two main veins here that are returning the blood back from the systemic circuit, the superior vena cava, which is up at the top, and the inferior vena cava down at the bottom. You're going to cover this in lab. From there, the blood is then going to go through the right AV valve or the tricuspid valve into the right ventricle, through the pulmonary semilunar valve into the pulmonary trunk arteries on the way to the lungs for gas exchange. Once the gas exchange has occurred, the blood is going to be higher in oxygen and lower in carbon dioxide. It's going to return back to the left side of the heart by way of pulmonary veins into the left atrium, through the bicuspid or left AV valve into the left ventricle. From there, it will go through the aortic semilunar valve into the aorta. Vessels coming off of the aorta then will lead to different parts of the body. So the valves make sure that the blood flows in one direction as it goes through the heart. And the valves open in response to pressure gradients. They're like swinging doors. So when the pressure in the atria is greater than ventricular pressure, the AV valves will swing open. When the pressure in the ventricles increases and is greater than atrial pressure, the AV valves will close. Ventricular pressure has to be greater than the pressure in the arteries, the arterial pressure, in order for the semilunar valves to open. As the blood flows through the semilunar valves, then the pressure in the ventricles will decrease and become lower than arterial pressure. So remember, the valves open in response to pressure, like a swinging door. And they should not open backwards, and we'll talk about anatomical structures that will prevent this from occurring in lab. The pressure of the blood in the chamber is going to increase as the amount of blood in the chamber increases. It's just like putting more water in a balloon. The pressure in the balloon will increase as it pushes up against the walls of the balloon. Let's begin now with the cardiac cycle. The cardiac cycle includes all the events of one complete heartbeat. Before we move on to the cardiac cycle, let's look at some terms. The term systole refers to the contraction of the cardiac muscles. So when the atria contract, you are in atrial systole. When the ventricles contract, you are in ventricular systole. The term diastole means that the cardiac muscle is actually relaxing, so you can have atrial diastole as well as ventricular diastole. Let's look at the cardiac cycle. It's going to include several phases. I'm going to list the phases for you first, and then I'll cover what's going to occur during each phase. The first phase is ventricular filling, or mid to late diastole. The second phase is isovolumetric contraction. The third phase is ventricular ejection. And the last one is isovolumetric relaxation. Keep in mind this is a cycle, so when you go to the fourth phase, then you're going to go back to the first phase, and it just continues. So all of this is happening during one heartbeat. For each phase, I want you to pay attention to a couple of things. I want you to notice which valves are open and which ones are closed, how the blood is flowing from where to where, or is it flowing at all? Are the chambers in systole or diastole? I'm going to use this figure in describing what is occurring during each one of the phases. This is a great figure for the cardiac cycle. I encourage you to stop the video and read the bullet points. But let's start with the first phase, ventricular filling, up at 12 o'clock. During this phase, blood is returning back to the heart from the circuits. As the blood is returning back to the atria, pressure in the atria increases. And remember, as soon as the pressure is greater than in the atria than in the ventricles, the AV valves will open. So as the pressure increases in the atria, the AV valves will open. The atria at this point are still relaxed, so you have atrial diastole at this point. About 80% of the blood is going to fill the ventricles from the atria at this point. Just passively move into the ventricles. And then the atria contract, and this is due to the SA node firing. And when they contract, they push the remaining 20% of the blood from the atria into the ventricles. Pay attention to the position of the valves. The AV valves are open, but the semilunar valves, notice, are closed. Let's move on to the second phase, isovolumetric contraction. As the pressure in the atria drops due to the movement of the blood into the ventricles and ventricular pressure increases, the AV valves will close. So what you have now is a closed chamber. All four valves are closed. So the AV and the semilunar valves are closed. The ventricles are going to contract, and you're going to have what's called isovolumetric contraction because the blood can't go anywhere. These are closed chambers. So the ventricles then will be in systole. When the ventricles contract and squeeze up against the blood, this increases the pressure in the chambers, the ventricles. As soon as the ventricular pressure becomes greater than arterial pressure, the pressure in the arteries that drain the ventricles, the semilunar valves are going to open. And remember, the semilunar valves are the valves between the ventricles and the arteries that drain them. So that, as soon as that happens, you are in phase 3, ventricular ejection phase. At this point, the AV valves are still closed, the semilunar valves are opening, and blood is being ejected from the ventricles. Then we move on to the fourth phase, isovolumetric relaxation. What occurs here, as soon as the pressure in the ventricles drops, the semilunar valves will close. Once again, you're going to have a closed ventricular chamber. So notice all of the valves are closed here, the AV valves as well as the semilunar valves. The difference is that the ventricles are relaxed. They are in diastole at this point. I'll talk about the end systolic volume and the end diastolic volume a little bit later when we get into cardiac output. From phase 4, you're going to move back into phase 1. So what will happen here is during phase 4, we're actually in early diastole and for the atria. So the atria are relaxed, but blood is starting to return back to the atria. As the blood enters the atria, the pressure in the atria will increase, and this will open up the AV valves, and that is what's occurring during phase 1. So as soon as the valves open, the AV valves open, you are back into phase 1.